Drosophila, or the common fruit fly, is a species of fly that is extensively used in a laboratory environment to explore the concepts of genetics and heredity, as it is an observable organism with a high reproductive rate, a short lifespan, and ease of maintenance. Because Drosophila can produce a new generation of flies in approximately 2 weeks, it is possible to discover the mode of inheritance for various mutations, such as the vestigial mutation. As a result, the purpose of the experiment was to determine whether the vestigial mutation in Drosophila was autosomal or sex-linked, and whether it was dominant or recessive. This was done by performing a monohybrid cross between pure vestigial flies and pure wild-type flies, and comparing the phenotypes of the F2 generation to known Mendelian phenotypic ratios. Fruit flies with the vestigial mutation are characterized by mutated wings, which are small in comparison to their wild-type counterparts. According to Mendelian inheritance, diploid individuals possess a pair of alleles for any particular trait. During reproduction, one of the 2 alleles for each parent of the offspring is randomly selected to be passed on. As a result, in a monohybrid cross, there are 4 different possible combinations of alleles. Each possible genotype corresponds to a phenotype, and, from this, the Mendelian phenotypic ratios of various crosses can be determined. Because Drosophila are sexually reproducing diploid organisms, the null hypothesis is that the phenotypes of the F2 generation will match Mendelian phenotypic ratios, whether it be autosomal or X-linked. It was predicted that if the mutation were autosomal recessive, then the phenotypes of the F2 generation would give 3 wild-type flies for every vestigial fly. Conversely, if the mutation were autosomal dominant, then the phenotypic ratios of the F2 generation would give 3 vestigial flies for every wild-type fly. It was also predicted that if the mutation were X-linked recessive, then the phenotypic ratios of the F2 generation for the vestigial males crossed with the wild-type females would give 2 wild-type females to 1 wild-type male to 1 vestigial male. The phenotypic ratios of the F2 generation for the vestigial females crossed with the wild-type males would equally give 1 wild-type female to 1 wild-type male to 1 vestigial male to 1 vestigial female. If the mutation were X-linked dominant, then the phenotypic ratios would remain the same, except the wild-type phenotype would be replaced with the vestigial phenotype in these ratios, and vice versa. The experiment consisted of 2 treatment groups, the vestigial males crossed with the wild-type females, and the wild-type males crossed with the vestigial females. The controlled treatments were the pure wild-type and the pure vestigial flies, left to reproduce amongst themselves in their respective vials. Whenever flies were anesthetized or transferred to new vials in order to be analyzed, counted, or sorted, the vials were always held sideways. Additionally, new vials were prepared by mixing 1 part dry Drosophila medium with 1 part tap water until the vial was filled to approximately 1/4 of its total height. After the water was completely absorbed in the medium, a pinch of yeast was added on top. Using the methods earlier mentioned, 4 new vials were prepared. Next, pure vestigial flies and pure wild-type flies from control vials were anesthetized. 4 male vestigial flies, 4 male wild-type flies, 8 female vestigial flies, and 8 female wild-type flies were isolated in each their respective labeled vials. The flies were kept isolated for a week, allowing fertilized females to lay their eggs and thus become virginized. After a week, the 8 virginized vestigial females were added to the vial containing the 4 wild-type males, and the 8 virginized wild-type females were added to the vial containing the 4 vestigial males. They were then left to reproduce for a week. When the females had laid their eggs and the pupa were visible on the sides of the vial, the parent generations were killed, and the pupa were left for a week to hatch. Afterwards, the phenotypes of the F1 generation flies were counted for each vial. 8 females and 4 males from each cross were isolated and transferred to a fresh vial and left to reproduce for a week. When the new females had laid their eggs and the pupa were visible again, the F1 generations were killed, and the eggs were, once more, left for a week to hatch. Afterwards, the phenotypes of the F2 generation flies were counted for each vial. All results and observations were recorded, and the phenotypic ratios were then determined. Because the experiment yielded a small number of flies, the phenotypes of the F1 and F2 generations from Chiara Vargas' team and Daryl Anthony Chouinard's teams were pooled together to get a more accurate chi-squared calculation result. The phenotypes from the flies of the F1 generation were counted for both vials. As compiled in this table, the wild-type males crossed with the vestigial females had 78 flies, all of them wild-type. In the wild-type females crossed with the vestigial males, there were 28 flies in total, all of them wild-type as well. The phenotypes of the flies from the F2 generation were also counted for both vials. As earlier mentioned, the obtained results were pooled with two other teams. For the wild-type males crossed with the vestigial females, this gave 168 wild-type flies and 49 vestigial flies, which gave a rounded ratio of approximately 3:1. With 1 degree of freedom and a chi-squared of 0.001675, the corresponding probability was greater than 0.9. In the wild-type females crossed with the vestigial males, this gave 149 wild-type flies and 50 vestigial flies, which also gave a rounded ratio of approximately 3:1. With 1 degree of freedom and a chi-squared of 0.677419, the corresponding probability was greater than 0.2. Given these high probabilities, the null hypothesis that the phenotypes of the F2 generation will match Mendelian phenotypic ratios was accepted. Nonetheless, it is important to acknowledge that the phenotypes of offspring are decided by chance, depending on the random assortment of alleles during gamete production. Furthermore, vestigial flies have a higher mortality rate than their wild-type counterparts because, as their name suggests, their wings are rudimentary. The mutation was found to be autosomal, as supported by the fact that both vials had 3:1 phenotypic ratios. Had the mutation been X-linked, one of the vials would have a 1:1 phenotypic ratio. Finally, in the F1 generation, all flies were wild-type, also suggesting that the vestigial mutation is recessive. Because the parent generation consisted of a cross between a homozygous dominant individual and a homozygous recessive individual, all F1 flies were found to be heterozygous. This is supported by the fact that all F1 flies were wild-type. Because the F1 generation consisted of a monohybrid cross, the F2 generation had 3 different genotypes: homozygous dominant, homozygous recessive, and heterozygous. This is supported by the fact that F2 vials had 3:1 phenotypic ratios.